Good morning, students. Today we will study the sexual reproduction in Oedogonium. Oedogonium is the female reproductive organ in Oedogonium. In the monoecious species, Antheridia and Oogonia are present on the same filament, but in dioecious species, they are born on different filaments. Development of Oogonia is, however, similar to both in the Necrandias and the Nenandrias and the Macrandias species. Any vegetative cell with the apical cap may function as oogonial mother cell. It divides transversely into an upper oogonium and a lower suffultatory or supporting cell. The supporting cell may again function like oogonial mother cell and form another oogonium and the supporting cell. In such cases, two or more oogonia are formed in a row. Initially, oogonium is slightly wider than the vegetative cell, but at maturity it swells and becomes globose and its protoplasmic form a single uninucleated non-motile egg. The young egg is green in color due to the presence of chloroplast. At maturity, a small pore or transverse slit develops on the oogonial wall. Just opposite this slit, a hyaline area is present in the egg which is known as the receptive spot. The egg nucleus moves under the receptive spot before fertilization. In some species, prior to fertilization, a gelatinous material is extruded through the pore to facilitate the entry of the anthrazoids into the oogonium. Fertilization. The process of fertilization is similar in both the Macrandus and Nenandrus species. Anthrazoids are attracted chemotactically toward the mature oogonium. Usually, a single anthrazoid enters into the oogonium through the opening present on the oogonial wall. If more than one anthrazoid enter the oogonium, only one of them fuses with the egg to form a diploid zygote. Soon after fertilization, the zygote separate from the oogonial wall and secrete a thick three-layer wall of its own. A newly formed zygote contains chloroplast and hence green in color. But as it matures, there is an accumulation of reddish oil in the protoplasm and it becomes reddish brown in color. Germination of zygote. The zygote is liberated by the decay of the oogonial wall and settles down at the bottom of the pond. It undergoes period of rest. This dormant state helps in the tiding over the unfavorable period. The zygote germinates under the favorable condition. The diploid nucleus of the zygote divides by meiosis to form four haploid nuclei, followed by the division of the protoplasm into four segments. Each uninucleated segment metamorphoses into a multiflagellate zoospore or zoomeiospore. The zoospores are liberated after the rupture of the zygote wall and behave like asexual spores. They swim for some time and then become attached to the substratum by their anterior end. Germination of the zoospore results in the formation of haploid filament. In Macrandus dioecious species, two of the four zoospore formed inner zygote developed into male filament and the other two into the female filament. Thank you.